Hello everybody, this is Dr. Vishal Trivedi from Department of Biosciences Bioengineering, IIT Guwahati. So, we will start our discussion with the discussing about the electromagnetic spectrum. So, as you know that electromagnetic spectrum actually contains the waves of different frequencies or different wavelengths, and all these different wavelength waves have the different amount of energy associated with it. So, what we have? We have so if we plot the so what we have and what we are more interested is to see what are the wavelengths we have within the visible light. So, within the visible light which is actually present in the sun is what we have. So, we have the light which is starting from 390 and which is going up to the 760 nanometer. Within this we have violet, we have indigo, we have blue, we have green, and we have yellow, then we have orange, then we have orange red, and then we have red, and after that we have far red. Okay. So, all these different wavelength lights actually are exposing the pigments which are present within the chloroplast, and that they are using it for the making the chemical energy. Okay. But by doing systematic experiments, scientists have found out that the blue light is actually containing the is very photoactive in doing the photosynthesis, because it contains more energy, whereas the red light is also doing the photosynthesis, but it contains less energy. But as far as the efficiency is concerned, the blue light, the red light, although the energy is less, the maximum amount of energy which is present with the red light is being absorbed. So, it is the most efficient in terms of the photosynthesis, and whereas in the case of blue light it is the most active, but the as far as the efficiency is concerned it is least efficient, because the lot of energy which is associated with the blue light is being go into the waste. And this region of blue to red is known as the photoactive region, because this is the region which is participating into the photosynthetic activity or PAR, and why the blue light has the more energy and less red energy, and red light has less energy, because the energy is expressed by the expression known as E equal to h nu, which is actually the discovery by the Albert Einstein. And if I put the value of wavelength it can be expressed like this. So, the shorter the wavelength, so if you have less, you are going to have the more energy. So, the wavelength of the blue light is lower and it is actually in the range of 470 to 500, whereas in the case of red, it is in the range of 600 to 700. So, the energy which is associated with the red light is less compared to the blue light, but as far as the efficiency is concerned, it is more efficient for the red light. Now, the question comes, although the visible light contains so many different types of visible light contains the light of different colors. So, how the photosynthetic machinery is utilizing this energy, utilizing this light of different wavelength to catalyze the reactions to convert the photo energy into the chemical energy. And that question comes when we see the absorption spectra of different pigments, what we have discussed in the previous class. So, what is mean by the absorption spectra? So, what is mean by absorption spectra is that if you plot the absorbance of a molecule against the wavelength in a 2D graph. So, what you have to do for getting an absorption spectra of a typical fluorophore or typical molecule or the pigment is you take the pigment and put it into a cuvette. Okay. So, you make the pigment, you make the suitable solution, put it into a cuvette, and then you take that cuvette and put it into the spectrophotometer, and then ask the spectrophotometer to excite this molecule with the different wavelength light and tell me what is the absorbance. So, what will happen during this particular kind of experiment? When you are exciting the molecules, the molecule is taking up this energy, and what is there in the molecule to take up the energy? The electrons. So, once the electron take up the energy which is present in the particular wavelength, it goes from one energy state to another energy state, and that actually will reflect in terms of their absorbance. So, if you take the particular spectra, what you will see is a pattern like this. Okay? And this particular pattern is going to be a signature sequence or signature pattern for the particular molecule, signature spectra for a pigment. As long as the pigment is in the state of the pigment is not changing, and as well as the solvent is not changing, means if you are making a solution of chlorophyll into the methanol, the particular pigment will remain, the particular signature spectra will remain intact until you are dissolving the chlorophyll into the methanol. But if you dissolve the chlorophyll into ethanol or acetone or any other organic solvent, probably the pattern will change, because that actually has the shielding as well as other kind of effects. So, this kind of spectra gives two information. One, that at this particular wavelength, the molecule is absorbing maximum. This means in this particular wavelength, which is actually the lambda max, the molecule is going to be most efficient, means it is going to take up this particular kind of energy into utilizing that energy for its work. The second information what we can also get if we also take a fluorescence spectra is that whether this molecule is also transferring that energy to other molecules as well. So, let us take let us see the absorption spectra of different pigments. So, what you can see is the spectra of different pigments, and what we are going to discuss is about the spectra of chlorophyll as well as the accessory molecules. So, what you can see is the spectra of chlorophyll A. So, you can see that the chlorophyll A is actually absorbing maximally in the red region, whereas the chlorophyll B is maximally absorbing in the range of in the blue, and the other molecule, for example, the beta carotene, what you can see this orange color spectra, the beta carotene is absorbing maximally in the blue region, whereas the phycoerythrin is absorbing in the range of 500 to 600 with the peak at around 550, and that actually gives the clues about how the chlorophyll machinery, which contains all these different pigments, are actually utilizing them to drive the photosynthesis process with such a high efficiency. So, what happens when any kind of light, if any kind of light falls, for example, if you keep the plant in a blue color light or in a red color light, it does not matter, because what matters is that as long as these pigments are present within the chlorophyll, for example, if you put it into a blue light, what will happen is beta carotene is going to absorb that light maximally and then transfer that energy to the chlorophyll and provide the energy to the chlorophyll. Although the chlorophyll is absorbing maximally in the red region, but since you are not even if you are not providing the red wavelength, it will take up the energy from the beta carotene, which is present within the vicinity in the chloroplast, and run the photosynthesis process. Similarly, you can understand how the phycoerythrin, which is also accessory molecule, is also going to participate in the process of photosynthesis. So, all these molecules are actually being used to drive the photosynthetic process, and that is why they constitute a complex, and that complex is known as the light harvesting complex. So, let us discuss what are the components are present in the light harvesting complex. So, as we said, the light harvesting complex are going to have a series of pigments Okay. So, imagine that the sunlight is coming from this direction. So, sunlight is giving the energy. So, what you need? You need a coat of the accessory pigments, so that the molecule which are present within the core of this light harvesting complex should not get this high energy light. So, what you need is a layer of accessory pigment, and we discussed that accessory pigment means the pigment which are belonging to the carotenoids or the phycobilin, and then these are actually constituting the antenna. So, antenna's job is to receive the light, primary light from the sun, and then they will take up this energy and then transfer this energy to the core molecules. Okay. So, these are the core pigment. So, these core pigments, then they will receive this energy, okay, and ultimately these core pigments are going to transfer this into the photo center or the reaction center. If you remember our discussion about the chlorophyll, we have discussed that we have two reaction center. One is called as the photosystem 1 and other one is called as photosystem 2. So, accordingly we have two different types of light harvesting complex, and these are called as light harvesting complex 1, LHC 1 and LHC 2, and LHC 1 actually contains the photosystem 1 and LHC 2 contains the photosystem 2. So, what you can see in the PowerPoint slide that so as we discussed, we have two different kinds of light harvesting complex, light harvesting complex 1, light harvesting complex 2. So, in the light harvesting complex 1 and the basic composition or the basic machine configuration of the light harvesting complex 1 or the light harvesting complex 2 remains constant. So, means that they have the antenna pigments on the periphery, then they contains the core accessory pigments which actually will be on the center, and then ultimately they are going to have the reaction center which actually will absorb or utilize this light energy to with the help of the electron donor or the electron acceptors. So, in the light harvesting complex 1, it exists in two different forms. One is called LHC1P680 and the LHC1P730, and these two are actually collectively known as the photosystem 1. So, photosystem 1, the reaction center, this reaction center in the case of light harvesting gun or the photosystem 1 is made up of a dimer of chlorophyll A P700, and it has a antenna complex, this the top antenna complex where approximately the 70 to 80 chlorophyll molecules are present, and it also contains 10 to 12 molecules of beta carotene. So, beta carotene is actually competent enough to absorb very high energy light, and whereas the chlorophyll molecules are also present. So, that if in case the high energy light is not coming, the chlorophyll molecule will also participate in the absorbing the light from the sun. In addition, they also contains the vitamin K1 iron sulfur cluster containing molecules such as ferredoxin and the plastocyanin, which is required for relaying and running the electron transport chain downstream to the electron acceptors. And because the light energy is being used to do photophosphorylation reactions, the photosystem 1 is actually participating in the cyclic photophosphorylation reactions, whereas where it does not require the assistance from the photosystem 2, which is actually not the case for the in the case of photosystem 2. Similarly, the light harvesting complex 2 contains the photosystem 2 or the photosystem P680. So, it actually contains the chlorophyll, which is actually containing a reaction center of P680, and the light harvesting complex 2 is present in the grana, whereas the light harvesting complex 1 is present in the thylakoid membrane, which are close to the stroma. The reaction center in the case of light harvesting complex 2 contains the chlorophyll A with the reaction center P680. The antenna complex in the case of light harvesting complex 2 is consist of the 20 to 25 chlorophyll molecules, beta carotene and the lutein molecules. The photosystem 2 contains pheophytin A. This pheophytin A is required to accept the electron which from the P680 reaction center, and this photosystem 2 actually catalyzes the first event of photolysis of water, and that because of that it also contains the quinone as well as the oxygen evolving complex. This quinone as well as the oxygen evolving complex is very much required for photolysis of water to release the electron, and that electron actually participate in the electron transport chain. So, what happen is that in the photosystem 2, because of the photolysis of water, the electron is released, and that electron is being circulated from the photosystem 2 to the downstream photoelectron transport chain, and that actually will be the will help in generating the different types generating the proton gradient, and that proton gradient is required for generating the energy. So, let us so both of these molecules, both of these light harvesting complex are doing the one basic process, and what are that basic process? So, these two light harvesting complex 2, light harvesting complex 1 is doing two basic functions. One, they are receiving the light from the sun, okay, and then light harvesting complex 2 is splitting the water to release the proton as well as the oxygen. These protons are making a proton gradient, or they are actually getting into the stroma. This half oxygen is evolving as O2, and in this process it is also extracting an electron which actually runs throughout the light harvesting complex 2, and then ultimately goes to the light harvesting complex 1. Light harvesting complex 1 is also receiving the light from the sun, and then ultimately giving this electron to the last complex, and that light complex is converting the NAD plus to NADH plus H plus. So, what is actually happening? So this proton gradient is being used to generate the ATP with the help of ATP synthase. Okay. So, these two light harvesting complex 2 and 1 are connected by each other by a series of electron transfer from light harvesting complex 2 to light harvesting complex 1, and that is known as the photoelectron transport chain. Okay. So, in the photoelectron transport chain, the light is exciting the light harvesting complex 2, that actually lysing the water to release the electron, and that electron is going through the several electron carriers and then ultimately going to the light harvesting complex 1, and ultimately it is reducing the NAD plus to NADPH, and at the other hand these two protons which are coming from the water is also generating a proton gradient, and with the help of utilizing this proton gradient and with the help of ATP synthase, the chloroplast is generating the energy. So, let us see what we have and how the electron transfers from the light harvesting complex 2 to light harvesting complex in a sequence of reaction known as the photoelectron transport chain. So, as you can see on the given PowerPoint slide, we have shown the complex photosynthetic electron transport chain, where you have two complexes, light harvesting complex 2 and the light harvesting complex 1, and all these complexes are present in the thylakoid membrane. So, in the case of as we discussed, the chain this light photosynthetic electron transport chain started when a water molecule is being split into the oxygen as well as the proton with the help of the oxygen evolving complex present onto the light harvesting complex 2. So, the chain starts with the activation of P60. So, once the sunlight falls into the light harvesting complex, it actually excites the light harvesting complex P680, and that energy it utilizes to do the photolysis of water, which is done by the oxygen evolving complex, and that complex actually split the water and produce the electron. The electron then enters into the P60, the light harvesting complex P68 of chlorophyll, which contains P680, and then from P680 it is being accepted by another electron acceptor known as pheophytin, and pheophytin is having the identical structure as the chlorophyll A, except that the pheophytin does not contain the magnesium within the center. So, it also contains the tetrapyrrole ring, but that tetrapyrrole does not contain the magnesium within the center. Then from the pheophytin, the electron passes through the several electron acceptor based on the reducing equivalents or based on the activation potentials, and then ultimately it goes to the iron sulfur cluster containing iron cytochromes, and that cytochromes then generates the proton gradient, and then ultimately it is being given. So, from the photosystem 2, the electron passes through the plastocyanin, and plastocyanin is a mobile electron carrier. So, it handed over the so it can actually move along the lipid bilayer, and that and as a result it actually can carry the electron and then give it to the another electron acceptors, which are actually present in the light harvesting complex 1. So, then the electron which is being generated from the water by the light harvesting complex 2 reach to the light harvesting complex 1, then light harvesting complex 1 also being excited by the light, and then it pushes out that electron which it has accepted from the plastocyanin to the another electron acceptor known as the vitamin K1, and then from the vitamin K1 it actually goes through a series of the electron acceptors known as the ferredoxin or the iron sulfur containing proteins, and ultimately being given to a reductase on the other hand, and that reductase actually reduces the NAD plus to generate the NADPH, and that generation is actually is required, and this NADPH which is being generated within the stroma is used for doing the many downstream photosynthesis reactions. On the other hand, the protons which are being generated from the photolysis of water as well as the other proton which are also being accumulated within the lumen goes out of the ATP synthase, and that also generates the ATP within the stroma part. So, as a result of this photosynthetic electron transport chain, as we discussed, that the electron flows from the light harvesting complex 2 to light harvesting complex 1, and downstream it goes to the reductase, and that actually generated the NADPH, and this NADPH is used for running the electron, running the downstream chemical reactions, as well as the ATP which is generated by the end of the electron transport chain is also being done. So, as you can see in the given slide, there are two types of electron transport chain are happening. One which is actually happening in the light harvesting complex 2, and that is the, so there are two, one which is happening in the light harvesting complex 2, the other one which is happening in the light harvesting complex 1. These two can communicate with each other, and as well as you can see in the slide that so there are two types of photosynthetic electron transport chain. Downstream to that, there are two different types of photophosphorylation events are happening, and these two photophosphorylation events are either in the cyclic reaction or in the linear chain, and all these are actually participate in generating the ATP. So, the purpose of photosynthetic electron transport chain is to acquire the energy from the sun and then utilize this energy to split the water into the proton as well as the electron, and that electron actually flows with the different components which are present in the electron transport chain, and ultimately that is being used for two different purposes. So, let us see what are these different purposes. So, photosynthetic electron transport chain is operating within the thylakoid membrane for two purposes. One, it is actually using or producing the reducing equivalents such as NADPH. So, NADPH is like a reducing equivalent, and you can see that it is actually being produced when the electron is passing from the photosystem. The second is that it is also producing the energy in the form of energy by catalyzing the phosphorylation. Means it is catalyzing a phosphorylation reaction where it is using the ADP and PI to generate the ATP, and that ATP is being used for various steps within the downstream chemical reactions which are actually part of this part of the photosynthesis. So, the reaction which are collectively being used to generate or catalyze the phosphorylation reactions to generate the ATP are known as the photophosphorylation, and within the ATP within the photosynthesis or during the photosynthesis there are two different types of photophosphorylations are operating. These are cyclic phosphorylation or the acyclic phosphorylation. So, is of two types. One is cyclic photophosphorylation and number two is acyclic photophosphorylation. There are specific conditions. There are specific conditions for in which the cyclic photophosphorylation or the acyclic photophosphorylation is being used to generate the ATP. So, let us discuss what are these conditions. The different conditions required for adopting the system for cyclic photophosphorylation is as follows. So, cyclic photophosphorylation is actually a primitive type of photophosphorylation which is happening until the bright light is not there. So, in the absence of bright light means when the high energy photons are not present is not coming from the sun, the cyclic photophosphorylation is operating so that it actually can provide the low level of ATP to continue the chemical reactions. So, low intensity light. Number 2, I am sure within some time you will understand that not only the low intensity light, but also the light of a particular wavelength, the absence of a particular wavelength is also forcing the system to go for the cyclic photophosphorylation. So, if the light of the wavelength lower than P68 is not available, then the system goes for the cyclic photophosphorylation. So, light wavelength. So, if there is an absence of light wavelength which is lower than P68 is not there, then the system will go for the cyclic photophosphorylation. Number 3, in the absence of carbon dioxide. Okay. So, if there is no carbon dioxide or there is a very low level of carbon dioxide is present within the vicinity of the plant, then plant actually goes for the cyclic photophosphorylation to continue the production of very low level of ATP from the system. So, let us see what are the different reactions are operating within the cyclic photophosphorylation. So, as you can see on the given PowerPoint slide, the cyclic photophosphorylation is, as we discussed, the cyclic photophosphorylation is happening within a specific conditions. And what are these conditions? The conditions are that the absence of the low light intensity, the availability of the longer wavelength means the absence of the light wavelength which is lower than the P680, and the non availability of CO2. So, under these conditions the photosystem 1 actually comes into the picture, where it acquires the light from the sun, which is actually the light of longer wavelength, and that actually starts the processes to catalyze the cyclic photophosphorylation. And in this what you see is that the P700 is acquiring the sunlight and excluding the electron from the reaction center. And why it is called as cyclic reaction? Because the electron which goes away from the reaction center actually come back to the reaction center after the after going through the different electron transport donors and acceptors. As we discussed earlier, the phycocyanin, which is actually a mobile carrier. So, the phycocyanin actually gives the electron to the P700, and that and then in the presence of light, the electrons comes out from the reaction center, and then these electrons are being accepted by the vitamin K1 or the quinone. Subsequently, these electrons are going through a series of different types of electron donor and acceptors, which are actually ferredoxine, and then ultimately it reaches to the cytochrome B to F complex, and at this stage the cytochrome BF complex which then get activated, and this activated complex then combines the proton to the plastoquinine, and then plastoquinine utilizes this proton gradient to produces the ATP in the presence of the ATP synthase. And on the other hand, the activated complex also take up the electron and give it to the plastocyanin, and that plastocyanin again give this electron back to the reaction center P700. So, as a result, what you can see is that since this is a kind of a compromised photophosphorylation, where the ATP is not being produced in a large quantities, but this is like a kind of a survival mechanism under the condition where the light intensity is very low and photosynthesis has to take place, so that the plant could be able to produce a very small amount of food and could be able to survive in that kind of environment. And so because of this kind of photophosphorylation mode is more common in the photosynthetic bacteria as well as the other primitive organisms, which actually does not get very bright sunlight. And so apart from the cyclic photophosphorylation, we also have the non-cyclic photophosphorylation or acyclic photophosphorylation. In the non-cyclic photophosphorylation, is actually a regular mode of photophosphorylation, where the both the photosystem 1 as well as the photosystem 2 are working together in accordance to each other. And in this system because the both photosystems are involved, the non-cyclic photophosphorylation system is only operating when the light of the lower wavelength as well as the bigger wavelengths are available. And because of the specific electron donor mechanism, this kind of non-cyclic photophosphorylation is also called as the Z scheme of electron transport. So, in this mechanism, what happens is that electrons are excluded from the photosystem 2 when the light is falling onto the PS2, and the reaction center which contains the P680 in the chlorophyll is actually doing the first event, that is, it is photolysing the water to release the electron, and then these electrons actually goes to the first electron acceptor pheophytin, and the pheophytin is subsequently take up this electron and give it to the more number of series of electron donor and acceptors, and ultimately it give that particular electron to the cytochrome B and cytochrome BF complex. From the cytochrome BF complex, which is then get activated. So, from the activated complex, the plastoquinone take up the proton, and then that proton is being used with the help of ATP synthase to generate the ATP, and the cytochrome BF complex then utilizes the plastocyanin to transfer these electrons to the photosystem 1, and then photosystem 1 again excluded the electron with the help of the light, and these electrons are then again taken up by the vitamin K1, and then a series of ferredoxine complexes, and then ultimately they reaches to the NADPH reductase, and that NADPH reductase then utilizes these electrons to reduces the NAD plus to NADPH, and as a result what you will see is that in the case of non-cyclic photophosphorylation, what you will see is the generation of not only the ATP, but also the generation of NADPH, and both of these molecules are actually being required for downstream chemical reactions which are happening after taking this energy, and be responsible for producing the light. So, what you can see is these two modes of photophosphorylations are actually responsible for running their own specific functions. The photosystem 1 or the cyclic photophosphorylation is required for sustaining the life of a particular organism, whereas the non-cyclic photophosphorylation is more efficient in terms of catalyzing the conversion of ATP and PI to ATP, and as well as it is actually the main photophosphorylation reactions which are happening in the actively photosynthesizing cells, and as a result, because of non-cyclic photophosphorylation, it generates not only the ATP, but also the reducing equivalents. So, with this we have completed our discussion about the receiving the light in the chloroplast by the light harvesting complexes, and how the light harvesting complexes utilizes this light to process the electrons which are being produced by the water, and how this flow of electron is generating the ATP and NADPH, which is being used for the subsequent downstream chemical reactions. So, with this we will conclude our lecture here, and we will continue with our discussion about how this reducing equivalent as well as the ATP is being used in chemical reaction in the subsequent lectures. Thank you.